The topic for today is about Lake Izabal in Guatemala. My colleague, Jonathan Oberst-Farner, is a native of Guatemala, and he noticed that basically none of the lakes in Guatemala, or almost none of the lakes in Guatemala, have been looked at carefully for paleolimnology. And so he's made it like a personal mission to basically go through and take cores from all of them, as you'll see a little bit here in the talk, and focusing on all kinds of things. But Lake Izabal is the first lake that we started working on together. And I met Jonathan in 2019 at an AGU meeting. And he was interested in this Lake Izabal from Guatemala, which is pretty interesting because it has something like, we think about 10 million years of sediment within the lake. And so today it's a fairly shallow lake system, and maybe it was always a fairly shallow lake system. But because of my interest in these sort of very long records and my interest in diatoms, using them for paleo reconstruction, we thought it was a good fit. And basically, this is sort of the outcome of those preliminary work that we did on it. And as you'll see, we've sort of expanded it kind of a lot since then. So, oops, I gotta click on the right thing. This one. So for people who aren't familiar with diatoms and their use, fossil diatoms are actually one of the more valuable fossils from lake sediments for a number of reasons. One of them is that they're very common, and they have very excellent preservation potential as long as your pH does not exceed 9 or 10, basically. Diatoms are, of course, found in a very wide range of habitats, and you don't need very much material. So one of the reasons that a lot of my colleagues like to have me on a project where we're looking at lake sediments is they can usually hardly tell I've even been in the core taking my sample from it, because we really don't need a lot of material. Diatoms grow very rapidly. They have, from what we know, some fairly rapid evolution, and there's many species that are labeled cosmopolitan, so we can say something about them even in environments that may be new or novel environments for exploration. And of course, diatoms have very short lifespans, so we're getting reconstruction of whatever that environment that they lived in a fairly short time window, and often they can rapidly colonize environments. So the list of things that I'm giving you is sort of a list of how you might make an ideal tool for reconstructing paleoclimate from lake sediments. You don't need a lot of material, it's preserved, they're found everywhere. All of these things basically contribute to our ability to use them for reconstructing past events in climates from lakes. Just for a little background, I usually use these terms in terms of like how diatoms live in the environment. So I will refer to them as plankton or euplankton if they're basically always in the water column, and that's their primary way of living, is floating in the water column. And settling through the water column is basically how they work their way out of the system. So they have to basically replicate fast enough while the lake is mixing to stay afloat in the water column. And so they're typically found in relatively deep water. And that is normally the way that we sort of interpret plankton when we find it in lake settings. So if there's a lot of plankton and not a lot of benthic species, then we usually assume that the water is fairly deep. And a lot of my research focuses on reconstructing past drought conditions and lake depth. And so frequently this is sort of a primary way that we utilize them. Another broad group that I often separate out are what I call miroplankton. Some people refer to them as tychoplankton, but I actually think that their lifestyle is intentionally staged. These usually form sort of large, heavy valves, and they're commonly form colonies. And a consequence of this is they tend to sink a lot faster. And so they have to be in the water column when the conditions are right for them. And then when the conditions aren't right for them, they tend to want to be out of the water column. And so they can form sort of marine-like snow or sticky mats that basically will drag other organisms out of the water column as they basically make their way towards the bottom. And one of the things that we know about Aulicosyra, even though they're single-celled organisms, they're living in a colony and they can actually detect the silica concentrations in the surroundings around them, and they can respond to this by changing the length of the colony. And so many Aulacosira, for example, have two different types of linking connections. They either have these linking spines that hold the colony together, separation spines that actually let the colony separate. And so if they need to make shorter colonies in order to stay in the water column, they'll form separation spines and the colonies will basically split. So as they grow and replicate, they don't get too heavy or massive and fall out of the water column. And if they are done being in the water column, they can effectively use this to increase their mass and forcibly sink themselves out of the water column. And so a lot of the Aulacosira are primarily living in these sort of staged lifestyles where they're in the water column as plankton when they want to be in the water column. And when the conditions are poor for them, they intentionally remove themselves from the water column. Unlike other things that I would usually separate out as tychoplankton, which are basically just things that are accidentally in the plankton, I think that the Aulacosira have an intention, if we can personify the algae for that, in that their strategy involves them being out of the water column when the nutrient conditions are poor for themselves. Another broad group that I will mention is just the benthic diatoms. So these are largely diatoms that live attached to substrates. They can be plants, mud, rock, zooplankton, or even other diatoms. And they're limited by how much light penetrates into the water column because they're attached to some substrate. And so typically, these diatoms, because they have a raphe, are able to reposition themselves by crawling around in the habitat, attaching themselves to substrates that they may prefer. Light conditions or water conditions change. They may actually migrate in the very slowly, but migrate along these substrates that they can be attached to. And because they're limited by sunlight and how far the sunlight penetrates into the water, where it interfaces with some sort of a substrate, they typically only occur in shallow water depths. And so our normal assumption when I find benthic diatoms in a lake setting is that they represent shallow water environments, especially when they dominate over the plankton. And so those are some of the premises that we use to reconstruct things like past water levels. This is an example from my dissertation. This is diatoms from Foy Lake. During normal conditions, non-drought conditions, the lake is dominated by plankton. This is a Lindavia variety, and it represents the water when the water is 30 meters deep or something like that. Today, we find it in abundance living in that setting. And during drought conditions, what we find is that the assemblages from the sediment record actually look very different. Instead of being dominated by plankton, they're dominated by benthic species. And so I've used this to some effect when I reconstruct past conditions, suggesting that our transition from planktic to benthic species is largely driven by changes in lake level. So I'm going to use this part in our discussion today. I just wanted to make sure everybody was kind of familiar with that idea. The other thing that I would mention is that diatoms are very sensitive to a wide range of environmental conditions. So most of my audience is probably diatomist, but we know that they're sensitive to things like pH, and this is one of the first applications that people used for paleolimnology. They used diatoms to reconstruct the effect of acid rain in lake systems, for example, and used all kinds of different techniques to showcase how the pH in lake systems can change through time. And so their preference for different pHs at the species level, but also there are some broad genera that tend to sort of fall into categories where they generally assemblages that are dominated by them would be more representative of either low pH or high pH conditions. So I will talk a little bit about the effective pH. And the other thing that I would say is I mentioned that their fossil preservation is also largely controlled by the pH. So if we see species that are found where there's alkaline water, they're often partly dissolved or largely dissolved diatoms in those settings. So that reaffirms our understanding of their nutrient conditions and also the water quality conditions that they might live in. Diatoms are also sensitive, highly sensitive to salinity. And so for people who aren't familiar with this, there are some genera between freshwater systems and marine systems that overlap. But generally speaking, when I see marine diatoms, I immediately recognize them as being marine diatoms because I cannot figure out what genus they belong to very easily because I focus on freshwater diatoms. And so working at some place where you're at a boundary where the salinity can change very rapidly or where there's a strong gradient from one side of a system through time to another side of a system means that we're likely to see very different diatoms associated with transitions in salinity gradients through time. And so, again, this is something that we'll sort of focus on a little bit today. And so those are just some background applications for how we generally focus on looking at large changes in diatom assemblages through time and use them to reconstruct things like pH, salinity, depth. Okay, so the focus of my talk today is going to be about Lake Izabal in Guatemala. And Lake Izabal is a large lake system. And Guatemala is a place that's in tropical settings. So if you're not familiar with the Lake Izabal system, this is a map showing it. The Caribbean the Caribbean Sea is to the top here, top right of the figure. And that is Amatigua Bay, which is actually the ocean. So that's the Caribbean Sea out there. And the Dulce River is a connection between Lake Izabal and an adjacent lake, which is known as El Golfete, which is effectively just a sort of a wide river, more or less. But it's a wide river in the form of a lake, in that the water is constantly flowing through Golfete towards the ocean from the Dulce River, and then also it flows into the Dulce River. So it's the same river system on both sides of the lake. And then it feeds directly into the Caribbean Sea. Lake Izabal is very close to sea level. It's only 1.5 meters above sea level, which means on average, there is a hydraulic head that forces fresh water and terrestrial settings towards the ocean. But there are times when the lake level can be below sea level. And what it means is that the hydraulic head of the water from the ocean will push up through the Dulce River, up through El Golfete into Lake Izabal. And so it does get a mix of brackish water that basically comes into the lake. In the modern setting today, we can measure that there is some salinity associated with it that is temporarily, at least, brought into Lake Izabal. And so this actually makes for a really unique setting for us because it's close enough to the ocean that we can see sort of an influence of marine influx, but it's a freshwater setting. So I feel comfortable looking at the diatoms that are present in this lake setting. And it also means that not only are we looking at things like climate change associated with the history from Lake Izabal, but we're also looking at sea level change because if I lower the lake level through drought and I raise sea level, one of the things that I might expect to have happen is more seawater will come into the lake. So this is the general setting. In 2018 and 2017, my colleague Jonathan Oberst-Farner went to Guatemala and Lake Izabal, and they collected 5 sediment cores. These are from various places in Lake Izabal. You can see them here, and they have differing lengths and go back farther or shorter in time as a result of that in terms of how far into the sediment that they go. But it gives you some sense of also the topography. There's mountains on both sides of the lake system, and it's in a valley between these very tall mountain systems that are adjacent to it. And then these are the cores that we collected. And also, if you're not familiar with where we are, there's an inset here that's showing us basically where Lake Izabal is near the coast. Near the Caribbean Sea. So just to give you some context for what we're looking at, this is a general model. What we did then was we took the cores that were extracted from the lake. These are all 5 of the cores side by side, and you can see that there's some very strong differences in terms of the sedimentation types. So the base of all of the cores where we got all the way down to some interval were basically organic rich mud. And then there's a transition, which we have designated here, listed as a flooding surface, above which the beds become laminated. And so we can see distinct small laminations associated with all of the sediments above it. And there's a picture here on the far right of the diagram that shows you sort of typical organic rich sediments at the base. This is our flooding interval, and this is not the whole core. This is just one drive, but you can see that they start to become laminated and continue to be laminated for several meters above this. And then there's a transition to basically thickly bedded mud, and there's some intervals in here that in various conditions are slightly different. But we can see this transition in the cores that got deep enough effectively. We can see this transition occurring where they become laminated. And then we can also see this transition where they stop being laminated near the top. And then plotted adjacent to these are a feature of magnetic susceptibility, which we use to basically help correlate between the cores. So these jaggedy lines that are drawn here are showing differences in magnetic susceptibility, and you can see that they all have this transition associated with the upper half or more of the core being dominated by material that has more or higher magnetic susceptibility, and then a transition to low magnetic susceptibility that allows us to not only see a visual link between them, but also there's a sort of like a chemical signature or magnetic signature that we can actually see. And all the red arrows are representing different radiocarbon dates that we collected from the cores. And we mostly focused on core 5, which is the best dated of these that are here. But I just wanted to give you sort of like a breadth of the rest of the cores showed similar transitions. What we're seeing is not something that's just limited to one core. And as we move forward, I'm mostly going to focus only on core 5 because it's the one that I analyzed and the one that most of us analyzed when we analyzed the record. And but I think it's representative of what we see throughout the rest of all of the core systems that are here that are collected from within the lake. After we got a series of dates, what we usually try to do in paleolimnology is to try to create an age model, which allows us to interpolate or extrapolate ages for samples that we don't have actual material that was dated from them. So this allows us to basically provide a date for those pieces that are between them. And what you can see is that the upper part of the core, well, we know when it was collected, so that gives us a very hard upper surface that has to be when the core was collected. And then a lot of times what we do is then just take a series of samples where we found abundant carbon material from the core, and we can use this to create an age model. This is a 10 radiocarbon age models that are AMS dated. You can see this red one was actually thrown away because the date seems to be very old relative to the rest of the dates that are here, but we use the rest of the dates to create our age model. And throughout the rest of the talk, there'll also be these pink and green bands that move across the core. Here it's sideways, but it's because it's showing us the depth. Just so that you're aware of what those are, based on data that we have, we think that the pink zones are basically sulfate-rich eosinic conditions. And then, so those are low oxygen, high sulfide conditions. And then these green intervals, which we think are alkaline intervals where the alkalinity was quite high. And then you can also see the long-term transitions going from basically modern conditions down to the base of the core is roughly about 10,000 years. So we're looking at a Holocene record associated with this particular lake system from this one particular core. I mentioned that we think that the pink zones are sulfide, and the way that we determined this was by looking at the total sulfur in weight percent associated with different parts of sedimentary records. And so this is from core 5. You can see the blue over here is plotting the weight percent of total sulfur, and it's contrasted against the total organic carbon curve as a ratio. And it's giving us some indication of what we think are likely sulfide-rich intervals or eosinic intervals where the sulfide values were quite high, but we think that they represent anoxic conditions with respect to most of the lake setting. And so especially at the bottom of the lake, they're going to be elevated sulfides and low oxygen conditions, which we assume is probably linked together with some type of marine incursion, because that's where sulfides typically stem from in these settings at high amounts. And also the addition of salt water into the system basically creates what we call a chemocline, or a chemical boundary that doesn't let mixing happen, where oxygen can basically be mixed down into the water column deeply. And so we think they represent a time in the past where there was a strong chemocline. This is also supported by the fact that our magnetic susceptibility transfers basically from this interval where we have a drop in the sulfides, we suddenly see a spike in magnetic susceptibility. And this is because sulfides often, when they're found in the water column, can create anoxic conditions, which then cause the ferromagnetic minerals or minerals that have iron-rich components in them to basically get dissolved. And so they don't preserve the magnetic susceptibility because the iron is basically not in a solid phase. And so what we're seeing is a transition where we have basically these really distinct boundary associated with change in magnetic susceptibility that occurs in all of our cores. And we see a high sulfide component trapped in the sediment. So we interpret those as being marine incursion associated events. And then these other transitions, the green ones that we list as brackish, are based on what we think are transitions in alkalinity. So these are all chemical approaches that we've used to basically reconstruct stratification. So we can see during intervals where there's eusinic conditions, there's very limited lake mixing, and that lake mixing starts to increase. And then we see this sort of freshening of the lake system associated probably with meteoric influx of rainwater that's being mixed together with the salt water, letting the salt water basically flush out of the system. Because one of the issues is lakes are a depression on the landscape. And if we dump a bunch of salt water into them, the salt water has a higher density than fresh water that might come in. And so when it rains into the system or surface runoff comes into the system, what happens is we create a freshwater lens that usually sits on the very surface of that lake system. And then because it's tied to a river system, the freshwater is more likely the part that's basically going to be carried out of that system into the adjoining systems as we work our way towards the sea. And so we think that a lot of the saltwater basically gets trapped in the system as a result of this based on the geochemistry. So if you're not into geochemistry, you're just here for diatoms, and you think, well, that's cool. You have some model. What do the diatoms say? I'm here to provide that for you. But I wanted to show you that these transitions that we see are not just limited to core 5. They actually occur. We have this sort of eusinic conditions associated with the stratified units. That also is a support for the idea that it stratifies strongly and that there's a chemocline, because when you have anoxic conditions, you can't get any kind of bioturbation in the lake sediments, which means that it preserves the small scale lamination features that we see. When there's oxygen that reaches the bottom through mixing, typically those bioturbation activities of organisms that burrow around in the sediment basically disturb all of the laminations, and they cause the laminations to be destroyed. So we see some support from this from the chemical and from the sedimentological approaches, that it was strongly stratified in these intervals and sulfide rich. So I feel pretty comfortable just from these things to say that we think that that's probably a marine incursion that we're capturing in our lake record. But I also want to say that we have looked at the diatoms in these settings, and I think that it broadly provides a similar support. So I'm going to work my way through the diatom stratigraphy of core 5 from Lake Izabal. One of the things I want you to note is that there's a lot of things that are just listed as SP123 or whatever up at the top, or conferred species that I am not positive what they are. And part of that's because when we analyze this, we were trying to get just a general picture of how the diatoms sort of relate to what we see in terms of the transitions. And we're working on the taxonomy now. It seems like it's probably going to be a bit of a task, because there hasn't been a lot of diatom work that's been done in Guatemala. And so I'm not always positive what I'm seeing is whether it's something new or something that may be similar to a species that we see in other places in North America, for example. So I'll try to showcase some of these for you, but I also want you to focus on the types of species that we see in the bottom part of the record. There's a little bit of tychoplankton in terms of Staurocyra and Staurocyrella, and some benthic species like the Ulnaria, Eunotia, Diplones, Luticola, Forstulia, Salafra, like everything that's down there. We don't really see much plankton at all. It's dominated by benthic species. And many of these genera are things that we usually find coupled together in dystrophic or like bog-like settings where we have elevated, more acidic conditions. So like a lot of Eunotia together with a lot of Pinularia and Star anise and Frustulia typically are found in kind of like acidophile conditions. So my estimation from this basically is that I think that these are likely some sort of a wetland type of environment that is dominated by plants and a lot of organic matter, which kind of tracks with what we see in the sediment record, at least. So I want to take a little detour and talk a bit about these diatoms. This is some of the diatoms from that assemblage. As I mentioned, they're basically all benthic species. Some of them are probably friends that you might recognize if you're familiar with freshwater diatoms from North America. So they're largely freshwater species and genera that we see. Some of them I have not been able to put a name on at all. In some cases, I'm struggling even to get it into a genus. So we're working on some of those at the looking at them in the scanning electron microscope. But many of these, I think, are probably classic diatom types that that we would expect to find in shallow water settings. And so today, where the core was collected is actually 15 meters deep or so. And so the idea that we don't have any plankton in the system suggests that the water at the time was much shallower than it is today, or at least some combination of it was shallower and light penetrated very deeply into the water to allow the substrates that are at the bottom to be sunlit. So I would normally expect to see some plankton in a lake system, even if it was just a couple of meters deep. So it suggests to me that we're in some sort of a wetland, shallow water setting at the very base of the record. As I mentioned, there's some things that we recognize. So there are some things in the list like Cimbella timida, Cimbella aspera. You can find those on Diatoms of North America in terms of the website, although I don't know that they have SEM images for all of them, but I'm pretty sure that's what these are and based on what I've seen of them in the light microscope and the SEM. There's a diatom that I'm calling Echinella guatemalensis. It's it seems appropriately named. But the pictures that we have of guatemalaensis are actually a little bit wider than the species or the taxon that I see in our record. And so I'm not completely convinced yet that that's what it is, but it's pretty close, if nothing else, to what we would normally call Echinella guatemalensis. This is also on Diatoms of North America. Help me identify that. And then there's some things that I don't know what they are, not only based on what I couldn't find on Diatoms of North America, but I struggled to try to figure, I mean, I know what genus they belong to, but I struggled to find something that was similar to them. In every case, I put the genus name. This is obviously a Psalophora. It's got a canopia. It's got all the other features you'd expect to see in a Psalophora, but the width ratio and the outline are kind of unique in that it's not even 50 microns long. And it's basically kind of got the shape of Psalophora americana and these sort of really wide-looking diatoms that we see that are usually much larger. Here's an example of a little tiny star anise that I couldn't find a model that was a perfect fit for in terms of its outline, but has all the sort of standard characters of a star anise. It has a staros, it has a pseudosepta on the outside of the valve, and it's got uniseriate stria. So all the things that we might expect to see. There are some things that I was a little confused by. So this is obviously an Amphipleura. It is not the one that I've ever seen. In North America, I usually see pellucida. This one is not pellucida. It doesn't have the striae density. When I first encountered it, I was like, either my microscope has gotten a lot better and I can suddenly see the striae on an Amphipleura very easily, or it's much, much lower density in terms of the striae concentrations. And I measured it on the scanning electron microscope. I got values between 30 and 31, which don't seem to match anything from North America that's been identified. And the closest thing to it has striae density that are usually in the 26 to 28 range. And it has a different central sternum than this thing does. So I'm a little miffed at what it is. I'm just calling it Amphipleura sp for now. But this is a nice view of that. I also found some Cnidium species I had a hard time putting a name on. This is kind of like the outline that you'd expect from Cnidium iridis, but these are not the proximal raphe ends of Cnidium iridis at all. Here it is in the scanning electron microscope. You can see the outline is kind of the classic shape of it. Proximal raphe ends and outline together do not match anything that I could easily find for Cnidium for North America. It doesn't mean it's a new species. It may just have not been able to find an example of it, but is something that I'm struggling with. Also found this Olicosyra, which I could not for the life of me figure out what species it was. It looks pretty similar to Crenulata in terms of like from a light microscope perspective, but it has ghost striae at the top of the valve near the spines. The spines are not Italica or crenulata-like in any way. And but it does have this kind of like ring-leased internal shape of the valve that's pretty similar to crenulata. So strange areoli shape, ghost striae, the spine shapes, and the strange shape of the ring lease kind of makes it into a category where I couldn't find a species for it. So, but this is a relatively uncommon diatom in the base of the core found in the early part of the record. There's another example. This is an anomalies. For the world might start off looking like Anomalies sculpta to you. It's very large, like sculpta is. It has an outline that's kind of similar, but the apices are definitely different shape than sculpta, and also all of the sculpta that I've seen never have 2 rows of medial areoli. But every one that I've found in this, including all of my SEM images and all of my light microscope images, have 2 rows of areoli in the medial area surrounding the raphe. So I could not find a Anomalies that fits this for North America either. So possibly a new species. And also I found this really cool ulnaria. I can tell it's an ulnaria, but when we got it into the SEM, you can see that it is biseriate. It's not completely unknown for ulnaria to have biseriate striae, but every example I could find was from China. So all of the ones in North America are uniseriate. And I couldn't find anything that matched the outline and had biseriate striae that was in North America at all. So, and it didn't really look like any of the Chinese versions that have been published. So I'm still working on some of that taxonomy. This is what I sort of think that the environment looks like at this time. And this is basically from Lake Izabal, from the wetland areas that are adjacent to the lake today. It's got mangrove wetlands kind of settings with a bunch of plant material that are in them. That's a Jesus lizard, I think, that I took a picture of. But this is kind of the setting that I expect it to look like: a quiet water, shallow system, dystrophic kind of environment with a bunch of plant material and probably variable acid conditions in that setting associated with the geology of the basin. And to support this idea, if you actually just go a little outside of the lake basin, this is a giant wetland area that's basically the Bocas del Polachic, which is a natural park system that's a huge wetland. And all of these flat areas that are adjacent to the lake in this topographic view are actually wetland areas today. So I think basically if we just lower the lake system, we can expand the wetland that's out here closer towards our core area, which is core 5 was taken right here. And probably that's a similar type of environment to what we were experiencing in the past. It just means that we had to lower the lake level considerably from where it is. So the next part of the record is actually an interesting transition. We see this very sharp transition where there's a bunch of plankton that show up, and also the presence of terpsino becomes very common in terms of particles that I could find. They're mostly fragments and broken pieces of it. But this transition from dominantly benthic to dominantly plankton is also the type of diatom that we see. Initially, when I started looking at these, I thought they were Thalassiocyra because I couldn't see any features to put it into anything else, but I know that it has like sort of sectors on the valve face, which are characteristic of Thalassiocyra. In subsequent work that we've done, I've actually seen some that look a little bit better preserved, and I think they're the same species, and they actually are actinocyclus. There's rings of Rimiportula that go around the outside on the mantle, but I don't actually have any mantles in SEM. So I originally was just calling it Thalassiocyra, and I think in the published version it's called Thalassiocyra, but I think it's more like a Thalassiocyroid. It's probably actinocyclus. So that's mostly brackish water marine kind of organism. And it's found together in our sediment with a bunch of terpsino. I think it might be musica, but I'm not positive about that either, because mostly what I see are fragments of it. It's highly dissolved when I find it, and this heavy causti that makes up the pseudosepta of terpsino is often all that I find. I don't actually find the valve faces or anything. I'll just find a little piece of a terpsino preserved. And I think that's because the alkalinity is quite high in these intervals. And so they're dissolving the silica. We clearly see the actinocyclis is also dissolved. But we think this is a result of the marine incursion bringing a bunch of salt water into our system. If we move up towards the top of the record, what we see is that suddenly there's appearance of a lot of granulata and a lot of Aulacosira type diatoms, followed by another interval where there's no diatoms. So just to give you some idea of what that looks like, the samples look like this. This is literally a picture from the slide where it's basically all the same monotypic, probably granulata or something very similar to granulata. Based on what we see in the SEM, you can see there's still some dissolution going on in some of these intervals. But this is what that sample looks like. The next samples that are above them, they look like this. There's nothing in them. So probably we dissolved out the remaining diatoms in the setting. It's not to say that they didn't exist then, but probably they're just not being preserved. And then the top part of the record, again, returns to this sort of dominance by Aulacosira. But one of the things that I would note is that we start to see a much wider range in terms of the type of plankton that are present at the top of the lake. And I think that this diversification of the plankton probably has something to do with elevated nutrients and some cultural eutrophication associated with the lake system. But we're seeing things like some sort of quilensis like Cyclotella, a bunch of different types of Aulacosira that are present in the lake system. And then there's also Discostella and some other clearly euplankton and meroplankton species dominate the upper part of the record. So these are freshwater diatoms again that we're seeing, and you can see the preservation of them is actually quite well in the SEM. So they're probably representing a freshwater setting. So I think the story that we get from this is that prior to 8,200 years ago, there was a wetland present here, shallow conditions, and then around 8.2, which is a sort of magic number in climate history where the Atlantic Ocean was flooded by a lot of water, probably led to a precipitous rise in sea level. And because the lake level here was low at the time, it means that a bunch of seawater flowed or inundated into the lake system, and then basically made the system eusinic for about 3,000 years after that. And it took that long before we could basically reconvert it back into a freshwater system, but it was still pretty alkaline in that setting. And then we see a return of diatoms in the preservation, and they're dominated by freshwater plankton from there to basically the top of the record, with one other interval that probably represents another marine incursion of some type that occurs between 1,000 and 2,000 years before present. So this is the summary of all of our data. I would point out that we have other things that people have looked at. These are examples of Cladocera that are found in the lake system. During the marine incursion, no Cladocera are really present. And then during these alkaline periods, there's no Cladocera preserved. But during the freshwater pulses, we see lots of Cladocera, and also the Cladocera types become more diverse as we move towards the top, the same story that we see from the plankton and the diatoms. And also testate amoeba have been looked at in this system. And what we see is the freshwater system, the most recent one, is the only one where testate amoeba are largely preserved. And if you don't believe that it's a marine incursion, that guy right there is a marine foram that we see at the place where the marine incursion event, where we think it occurs. So only one likely explanation for that is it's probably being carried into the system from the ocean. So generally, this is our story for what's going on in terms of the history for Lake Izabal. We see the early part of the record dominated by benthic diatoms, a flux of marine-like conditions in there that changes it rapidly to plankton. And then progressively, it becomes less and less dominated by these sort of eusinic conditions, becomes more alkaline in nature. And then the top part of the record, two major intervals of freshwater diatoms separated by this sort of alkaline zone. So clearly, sea level influx has played a role. One of the reasons that we think that this research is actually pretty important is because there are a lot of places that are near coastal environments where there's freshwater settings that could be inundated as sea levels continue to rise. And so there's a conceptual concern here, which is that in the system where we saw a substantial change in sea level where the lake was inundated by marine waters, at least from what we saw, the diversity in those systems in terms of what's present in the plankton, in terms of the diatoms and other types of organisms, are all dramatically affected by this sort of pulse of marine waters that can come into these systems. And there's a fairly large part of the world that could be strongly affected as sea levels continue to rise, and basically are threatened ecosystems. And we start looking at the long-term consequences of ecosystem changes. These these environments that are basically adjacent to the ocean are particularly endangered by this potential conditions, which could potentially cause chemical changes that last in those systems for thousands of years, at least if Izabal is a indication of what we see. Some current research that we're doing on Lake Izabal. So my colleague Jonathan really loves coring lake systems. We went back to Lake Izabal and took 26 cores from Lake Izabal and the surrounding El Golfete to try to see if we could get a better handle on how much the salinity and alkalinity in the system and that sort of chemocline system was basically in place. The red dots are ones that are long core records that we're currently analyzing, and I'm also going to analyze one of these cores from here near the wetland area so I can get a little bit of the modern diatom diversity, because a lot of these are in deep water and so they're mostly dominated just by plankton. So I want to try to get some idea of what we're seeing in terms of the transitions from more shallow water settings and try to see what we can recover. But the blue ones are basically about 1 meter long cores that we have from all over the lake system. These were collected two springs ago. Jonathan was there in Guatemala sampling stuff for a long while. Another aspect of this that's actually kind of interesting is that the placement of Lake Izabal is actually right on the boundary between the North American, Caribbean, and Caicos tecton plates. And there's a lot of tectonic activity associated with it. We see areas where there was massive amounts of earthquakes associated with the 1816 Polachik earthquake. Right here is outlined in blue. And, the Izabal area is in this sort of dark red area here. That we think might be strongly affected. And Guatemala was heavily damaged by earthquakes in 1976 in the Montagua earthquake that killed a lot of people and caused a lot of damage associated with Guatemala City. But this is the Montagua Fault, and it is right adjacent to Lake Izabal. Also plotted on here are a whole bunch of other lakes. This is Ayarza, Amatitlán, Atitlan, and here's Izabal, Chicho, and Lechua. And Jonathan's actually collected cores from all of these lake systems because they were there for the entire spring, 2 springs ago. For months, they were coring. But what you can see is actually in all of these lake systems, except for Izabal, there's a distinct earthquake deposit that we've been able to recover. So here's Atitlan. Earthquake deposits are in gray. Here's Lake Ayarza with gray cores collected that show the earthquake deposits. Here's Chicho with the same thing. Here's Lechua with the same thing. And here's an earthquake deposit from Amatitlán. There's actually another one down at the base of that one. And you can see just if we took a series of cores from Lake Atitlan, that there's a whole series of the earthquake deposits that occur across the entire system, this sort of unstructured giant sediments that influxed into the lake system during the earthquakes. And here's another one from Lake Ayarza, where you can see all of the earthquake deposits from Ayarza linked from one core to the next across the entire lake system. So significant influence of earthquakes in these systems. And of course, we can look at the diatom records from these. I've had graduate students who've been working on the stratigraphy of many of these lake systems for the diatoms. And one of the things that we see is during earthquake intervals, there is some sort of transition associated with a response in terms of nutrients that flux into the lake. So as we shake a bunch of the landscape and we flux a bunch of the sediments, we're also bringing nutrients into the lake system. And here in Lake Atitlan, you can see there's a very strong transition. Suddenly we get Alloxyra granulata at the top, where previously it wasn't present in the record before the earthquake. And so we think this is tectonically forced eutrophication of the system. And if I move on and talk about Lake Ayarza, we also see a similar event. It's dominated by Cyclotella. And then at the top, basically a sudden shift in terms of the species that we see. And also we move away from all of these sorts of benthic species into plankton because of the probably loss in water clarity associated with elevated nutrients in the system. So this is a sort of interesting transition. We're looking at a bunch of these records to try to see if there's more examples of tectonically driven eutrophication events associated with these events. This is the 1976 earthquake. And this is our record from Lake Izabal. What you don't see is a very clear layer that's the same across all of our cores. So there's actually no clear observed widespread indication that the earthquakes are playing a direct role in terms of the eutrophication of this lake system, at least that we've been able to detect so far. But we're going back to these cores and we're looking at them at a higher resolution, and we're trying to sort out the taxonomy a little bit better. So these are some of the things that we're still working on this this project. And that's my talk. So hopefully you enjoyed that.
Great. Thank you so much, Jeffrey, for a really interesting talk. Maybe oh, what are these? Some trumpets coming up on the screen. um,
There's trumpets.
yeah, I just saw a trumpet go. Or what is that?
Oh,
We got
oh,
thumbs
it's a
down,
celebration. Maybe they're
celebration.
cel. Yeah, maybe they're celebrating. They're glad
Um,
it's over.
there we go. Mark has a question.
Yes, Mark.
What do you got, Mark?
I gotta turn on some cameras and stuff here. There we go. There we go. Oh, I'm back. I'm back. Quick, Jeffrey, what's the origin of this lake? I mean, you started getting to the suggesting there was some rifting and stuff going on. Is that
Mhm.
truly the origin of a 15-meter deep lake in
It is.
point 0.5
This is
1.5 meters above sea level?
Yes, this fault system that's here is actually kind of an interesting fault system. It creates a set of faults that bound both sides of the lake. And as I mentioned, there's something like a 10 million year sediment record into Lake Isabel, or what we think is 10 million, like 10 million years of lake sediments that go in there. And that fault is probably the origin of it. And so what's interesting about it is that I don't think that the basin has ever been very deep. It's basically just subsiding very slowly, and it's keeping
Mhm.
up more or less with sedimentation rates. So I don't know that the lake has actually been, at least from what Jonathan has told me. So he's actually a person who studies faults and sedimentology. I mean, I'm a geologist, but don't let them know that, because they still think I just do diatom stuff, and I'm happy that way. But the fault system that's there, that they think that that's just been slowly down dropping, and it's basically just kept the lake at this elevation. One of the things that's kind of cool about it, so we have a project called Libre, which is meant to try to core back 10 million years into Lake Isabal, and Jonathan's leading that project. And part of the reason I'm super interested in this is because I want to look at a record of 10 million year diatom record from lakes from the tropics, because there's nothing like that anywhere. And I only have both projects associated with both Tanganyika and Isabel, both have these sort of 10 million year long records, but we haven't been able to get the funding to collect cores that go back that far yet. So I'm tiptoeing around it, hoping eventually we'll get funding for those to actually get a full
So,
very, very long record from these settings.
so that lake could be that old. That's crazy. Another
It
quick.
almost certainly is. There's well
Okay.
data
Okay.
that
Okay.
goes back
Okay.
10 million years. So
Wow. Other quick question is, with these coastal systems like this, did you see or do you see any sort of indications of hurricane or typhoon impacts on these systems?
I haven't seen anything that I would recognize as a tempest-driven deposit in this setting.
Okay.
The Isabel's pretty far inland from it, and it actually gets really heavy rainfall like all the time. So
Mm-hmm.
maybe, and it mixes pretty frequently today. So maybe it's hard for me to see what a storm would look like, because it's always storming there.
Sure.
But
Okay.
some of these other systems might actually experience that. The record from Amatitlan is actually super interesting. It's got about 25 meters of core, and it only goes back about 5,000 years, based on the date that we
Wow.
have. So there is an incredibly high resolution record that's coming, potentially coming through there. And I have a undergraduate who's going to become a PhD student working with us, Mahalia, whose research is focused on that record, and we're probably going to try to get the whole 5,000 year done at very high resolution to try to see what we can uncover. And that's probably our most promising story for tempestites, is something. It's also along the coast. It's the other coast, but it's along the coast, and I think that that kind of a resolution would actually be capable of detecting storm activity a little bit better than what we see from Isabel, which is lower resolution in terms of time.
Cool. Neat. Thanks.
Hey, Anna had made a comment about a basilisk, probably the most important component of a wetland.
The basilisk is this lizard that we saw in the picture. There we go. There
Ah,
he is. He's
okay.
smiling. He's smiling at us. He ran across the water before he stopped on the lily pad, and I didn't get any pictures of him running across the water. But yeah,
Yeah, yeah. And Layla, do you want to ask your question about marine incursions?
Yes, thank you. In fact, your marine intrusion, you said, of course, you had the foraminifera, you have the change of the mineralogy in the base of the core 5, but you base it mainly on Telpsinoe. But Telpsinoe, some people think that it's more freshwater, or for me, I found it just in brackish water. So, but I'm agree it might be marine too. But you have Ketoceros, and we don't see it. We see it up. What about it?
So there's just a little bit of Catastrophe cyst near the boundary where the it's actually like just this little tiny piece here.
Yeah.
And then we see it occasionally. I see some cysts that are basically present in the upper part, but it's such a small fraction. I didn't really focus on it. But I would actually agree with you. I think Terpsinoe is probably brackish water conditions. And that's the way I sort of interpreted it here. But I think that the actinocyclus is probably also heavily, more marine-like conditions. And also there's a sudden jump from benthic diatoms to plankton, and it's timed perfectly with this sort of flux of high brackish conditions. So the only way that I could really perceive that happening is we dump a bunch of ocean water into our lake system to get it to rise that quickly, and then also to transfer into brackish conditions. When it's shallow, it's fresh, right? So that's kind of interesting. If it was shallow and salty, that would be a different story, but it's shallow and fresh. And so it's shallow and fresh to deep and salty, or deep and brackish, suggests that we had to have some kind of influx of salinity in order to produce that, in my mind.
So your change, do you explain it with the climatic change or with seismic as well?
I think it's all driven by the pulse. The 8.2 event's a very substantial event in terms of marine history, that we have a pulse of freshwater that comes out of the North Atlantic and starts to basically fill up the Atlantic Ocean a little bit more. And it doesn't actually take a lot of change in sea level for us to go from dumping stuff into the ocean for the ocean to then be dumping stuff into our lake system. So even a meter change would be enough to reverse the head in the system. And especially if the lake's already low, all we have to do is get back over the freshwater sill into the lake system, which probably doesn't require very much elevation. So I think it's being driven by that, largely because of the timing. So if it were any other time, I would say I'm not so sure, but rated 8.2 makes me very suspicious. And so I think it's probably a rise in sea level associated with it. I mean, you could argue that it's tectonic, but the problem with the tectonic explanation is it doesn't get the sea into the lake. It actually just makes the lake either lower or higher, right? So it doesn't give us a good explanation about how we would get sea into the lake system.
And you are sure, and you don't have any discontinuity, any loss of sediment in your core?
Not based on the record that we have. And if you look at the age model here, wherever I plotted that age model, there's actually kind of a lot of dates around the 8.2 kind of time frame. And I'm not sure, I mean, this is maybe a little sketchy here, but these transitions don't show a major hiatus associated with it. So I would say no.
Thank you very much.
Sure. Thank you for the questions.
Yeah, I have a question, Jeffrey, about, so you've shown, this is sort of you have a region with a few lakes in it, and they seem to show that same seismic signal across them. What else joins these lakes in response in some way? Sort of, are there any like regional climatic signals that are common across the lakes, or anything else like that makes them coherent as a sentinel?
I would say this is our task currently. So I don't have an answer to that question, but we also we don't have much of a climate record from Guatemala. We have Pizza and Itza, which is, far on the coast. It's the closest sort of long-term record that we have a record from. And then all of these lakes, we really have only looked at the Holocene record from Izabal, and the other ones have like 1 meter records that we've looked at that are probably, 1,000 years or maybe 2,000 years at most. And so, and we definitely haven't reached the point where I feel like we could look at coherent signals between them, but that's our goal, right? If it's a climate story, so Layla asked that question about, could it be a climate or tectonic influence? I think the only way that we can address that is by having enough regional stories to actually see what the climate is like. So if we can get back 9,000 years ago and see if there's a drought associated with our, freshwater low stand, that's going to be okay. That'll be like a way of linking these things together. Sea level probably doesn't affect all of these lakes because many of them are inland and have no connection to the river. And earthquake story is actually kind of a cool one. So Jonathan's just publishing that in Geology in a couple of months. I think it'll come out. But you can kind of see why Izabal isn't really affected so much. It's actually behind the focus point, and most of the directionality is actually towards the other lake systems. So like if the focus is here, and then all of the seismic energy is sort of shooting to the west of there, Isabal's not really probably going to get a lot of shaking associated with that. And so we think that's probably why we're not seeing much of a signal from Izabal, or not as much of a signal. Chicho, we don't have a very good signal from either. And Lechua doesn't have any diatoms preserved. So like these are our good targets, and they're the ones that we've looked at the most closely. And we see like a clear seismic response from those three.
Okay. Well, we're at the top of the hour, so I want to thank you again, Jeffrey, for sharing your work on this interesting part of the world, and thank everyone for joining. And I also just posted, Jeffrey also runs some really good, fun resources online on his Diatom Attack. Diatom's Attack. Would you call that a channel, or there's I put in the Twitch, the Instagram, the YouTube, and the Redbubble. So you can check out the great stuff that Jeffrey does in his free time.
Yeah, I like doing outreach for science, especially associated with diatoms, and I like to show people what scientists are actually like and what they're doing. So I do a lot of outreach, social media type stuff associated with that. So
that's awesome. Thank you so much for taking diatoms to the world.
it's part of our job.
It's part of our job.